Welcome back to Sleepy Science After Dark, where we explore the quiet mysteries of biology and the slow, patient march of time itself. Tonight, we're settling in for a gentle exploration into something that touches every single one of us. Something that's happening right now, at this very moment, in the billions of cells that make up your body. We're talking about aging. It's one of those universal experiences. Every living thing that exists long enough experiences it. Plants age, trees age, fish age, whales age, and we age. Now, aging might sound like a simple thing on the surface. You get older, your hair grays, your skin wrinkles, and one day you can't quite remember where you left your glasses, or your car, or what year it is. But beneath that simple surface, beneath those obvious changes we can see in the mirror, aging is one of the most fascinating, intricate, and deeply compelling biological processes we know of. It's not just decline. It's not just time passing. It's a coordinated symphony of cellular events, a cascade of changes written into the very fabric of your DNA. It's an orchestra of processes, all playing slightly out of tune. As always, I hope this video helps you drift off gently into sleep, or at least helps you see your own body with a little more wonder and appreciation. There's something oddly soothing about understanding that every wrinkle, every grey hair, every ache and creak is a story your cells are telling. It's not malice, it's not evil. It's just the inevitable consequence of being alive for a long time. If you haven't already, I'd love it if you gave this video a like and maybe subscribe to join our growing community of curious, sleepy minds. We're building something special here, a community of people interested in understanding the world a little better. Now settle in, get comfortable, maybe grab a warm beverage, and let's talk about why we age. Let's explore the mechanisms, the processes, the elegant and heartbreaking details of how our bodies change over time. So what exactly is aging? It's a question that sounds simple until you really try to answer it. At the most basic level, aging is the progressive deterioration of biological function over time. But that definition, while technically accurate, doesn't quite capture the full picture. Aging isn't just about things getting worse. It's about change. It's about transformation. It's about your body gradually becoming less able to repair itself, less able to maintain the delicate balance we call homeostasis. There's a word for that balance, that state of equilibrium that your body tries to maintain. It's called homeostasis. And as you age, your cells and tissues become steadily worse at maintaining it. It's like your body has thermostat, a pH sensor, a blood sugar monitor, all these little systems trying to keep everything within safe ranges. Your body can't cool itself as efficiently when it's hot. The cooling system, your sweat glands and blood vessel dilation, work less effectively. It can't generate heat as effectively when it's cold. Your thermogenesis machinery sputters. It can't maintain steady blood sugar or regulate blood pressure quite as smoothly. The feedback loops that kept everything in balance start to have delays, start to overshoot, start to respond too slowly. Little by little, year after year, your body becomes less resilient, less flexible, more fragile. The margins of safety shrink. What wouldn't bother a young person becomes dangerous for an older person. Your cells are like a factory that's been running for a very long time. Initially, when the factory is new, the workers are energetic, the machines hum smoothly, and everything gets repaired almost immediately when something breaks. A machine starts to wear down. A technician notices and fixes it immediately. A worker gets tired. They take a break and recover. There's redundancy built in. There are backup systems. There are checks and balances. If one assembly line fails, another takes over. If one enzyme stops working, backup enzymes can sometimes compensate. But as decades pass, the workers get tired. The most experienced workers retire or slow down. The machines start to wear out. Parts become harder to replace. The redundancy gets used up. Repairs take longer and longer. Eventually, the technicians themselves get tired and less efficient. And then, at a certain point, something critical breaks. And when it breaks faster than it can be fixed. That's aging at its core. It's not dramatic. It's not a sudden collapse. It's a gradual tipping of the balance from repair exceeding damage to damage exceeding repair. The curve slowly bends, the trajectory shifts, and you cross an invisible threshold. But here's the thing that makes aging so fascinating from a biological perspective. It's not random. It's not chaos. There are patterns to it. There are mechanisms. There are actual clues written into your cells about how and why aging happens. Some of these mechanisms happen in the nucleus, deep within your DNA, that coiled blueprint of instructions. Others happen in the mitochondria, those little power plants of your cell that generate the energy needed for everything else to work. Some involve inflammation, oxidative stress, and damage accumulation, a kind of biological rust spreading through your tissues. Some involve the shortening of protective caps on your chromosomes, a molecular timer counting down. Some involve cells simply deciding to stop dividing and enter a state of permanent quiet, like workers clocking out for the last time but never leaving the building. When you look at aging this way, not as a mysterious force, but as a collection of biological processes, it becomes less frightening and more interesting. You're not witnessing some vague decay. You're watching your cells navigate a complex landscape of damage, repair, adaptation, and ultimately exhaustion. And that process, that long, slow waltz between what your body can fix and what it cannot, that's what we mean when we say aging. Now let's talk about one of the most elegant discoveries in modern biology, something that sits at the heart of how cells keep track of time. It's called the telomere, and if aging has a clock, this is it. Picture your chromosomes, those X-shaped bundles of DNA inside almost every cell in your body. At the very ends of these chromosomes, like the plastic tips on shoelaces that keep them from fraying, are structures called telomeres. They're made of repetitive DNA sequences, T, TA, G, 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 repeated over and over and over again, thousands of times. This sequence might seem random, but it's one of the most important pieces of biological machinery you possess. These repetitive sequences protect the actual important genetic information at the ends of your chromosomes from getting damaged or lost. If telomeres weren't there, the important genes at the chromosome ends would get damaged with each division. Each time a cell divides, each time it copies its DNA and splits in two to become two daughter cells, something unavoidable happens. The telomeres get a little bit shorter. Not much, usually about 50 to 200 base pairs, but consistently shorter. The count goes down. The sequence T, T, A, G, G, G appears fewer times. This isn't a design flaw. This is actually a feature, a built-in counting mechanism. Evolution designed it this way. You can think of telomeres as a cellular counter, and every time it clicks down, your cell is keeping track of how many times it's divided. It's like a roll of toilet paper that gets a little shorter each time someone uses it. Or like the candles on a birthday cake getting fewer each year. Eventually, it runs out. After a certain number of divisions, usually somewhere between 50 and 70 times for human cells, the telomere becomes so short that the cell essentially says, enough is enough. This is called the Hayflick limit, named after the scientist who discovered it. When a cell reaches this limit, it stops dividing. It enters a state called senescence, which we'll talk about more in a moment. But first, consider the elegance of this system. Why would your cells have a timer built in like this? Well, there's a very good reason, and it's worth taking a moment to appreciate the evolutionary wisdom here. Cancer cells are, at their heart, cells that have broken the rules. They keep dividing when they shouldn't. They ignore signals to stop. They acquire mutations that let them bypass normal safety mechanisms. Imagine if a cell could divide infinitely. If it had no limit, it could accumulate mutations with each division, gradually becoming more and more abnormal, eventually becoming something genuinely dangerous and cancerous. Evolution faced a problem. On one hand, you need cells to divide so you can grow, repair injuries, and replace old, worn-out tissues. On the other hand, if cells could divide forever, they'd eventually become cancerous. So evolution came up with a solution. A clever solution. Telomeres act as a brake. They make sure normal cells can't divide forever. After a certain point, they simply can't anymore. No matter how much energy a cell has, no matter how many growth signals it receives, once the telomeres are gone, the cell cannot divide. It's locked in. The telomere is a biological insurance policy. Each cell gets a limited number of divisions, and that's it. That's the trade-off, a compromise between growth and cancer prevention. But like all insurance policies, it comes with a cost. That cost is aging. Your cells age because they have a limit on how many times they can divide, and this limit exists specifically to protect you from cancer. It's one of nature's great bargains. You get to live your whole life without developing cancer, without rogue cells multiplying uncontrollably. But in exchange, your cells can only rejuvenate themselves so many times before they have to stop. Every time your telomeres shorten, you're watching one of the most fundamental processes of aging unfold at the molecular level. It's happening right now in your skin cells, in your white blood cells, in the cells lining your gut, in your immune cells, fighting infections. Tick, tick, tick. The countdown continues. Now, it's worth noting that not all cells in your body follow the Hayflick limit. Some cells, like germ cells and stem cells, have access to an enzyme called telomerase. This enzyme acts like a molecular fountain of youth for these special cells, a true fountain of youth. It doesn't just preserve telomeres, it actively rebuilds them, adding sequences back onto the telomeres so they can keep dividing. In essence, telomerase can reset the cellular clock. The telomerase enzyme can add new T, A, G, G sequences to the ends of chromosomes, restoring the counter to its starting position. Cancer cells have learned to reactivate telomerase, which is one of the reasons they can divide so many times. They find a way to keep that biological clock running. But in most of your regular cells, most of the time, telomerase is silent. It's not active. It's been turned off by evolution. So those cells age according to their telomeres, one division at a time, tick by tick by tick, until eventually they reach that Hayflick limit and the dividing has to stop. But aging isn't just about telomeres. In fact, telomeres are just one piece of a much larger puzzle. Because your DNA, that beautiful molecule that contains all the instructions for building and maintaining you, is under constant assault. Every single day, your cells face damage to their DNA. Think about that for a moment. Every single day, not once in a while, not occasionally. Every single day from the moment you're born until the moment you die, your DNA is being damaged. Some of this damage comes from outside. Ultraviolet light from the sun can damage skin cells, breaking chemical bonds and creating lesions in the DNA. Radiation from space can damage cells deep inside your body, causing double-strand breaks that split both strands of the DNA helix. Chemicals in your food and environment can insert themselves into your DNA or modify it in harmful ways. Chemicals called mutagens. But here's the thing. Most of the damage actually comes from inside. It comes from normal cellular processes. When your cells burn energy, when they use oxygen to power their functions, they create byproducts called free radicals. These are highly reactive molecules that have lost an electron and will do almost anything to get one back. In doing so, they cause damage. They can damage DNA, lipids, and proteins. They're like tiny wrecking balls inside your cells, bouncing around and smashing into things. Your body has defense systems against free radicals, molecules like antioxidants that can neutralize them by donating an electron and becoming stable themselves. Vitamins C and E are antioxidants. Glutathione is an antioxidant your body makes. But this defense system isn't perfect. It's overwhelmed. Some free radicals get through. Some damage occurs. And as you age, as the cellular defenses gradually weaken, more and more damage accumulates. This is called oxidative stress, and it's one of the hallmarks of aging. The balance between free radical production and free radical defense tips toward damage. Now here's the remarkable part. Your cells have repair mechanisms. They can actually detect damage and fix it. There's a system called nucleotide excision repair that can cut out damaged DNA and replace it with new, correct sequences. It's like your cells have a very sophisticated word processor with find and replace functionality. There's base excision repair that can fix smaller, more subtle damage, individual bases that have been modified by oxidation. There's mismatch repair that hunts down mistakes in DNA copying, finding and correcting typos. There's even a system called non-homologous end joining that can stick broken DNA strands back together, essentially using molecular glue to repair fractures. These systems are remarkably sophisticated. They work most of the time. They keep your cells running smoothly for decades. But like all machines, like all systems, they gradually wear out. The enzymes that do the repair work get less efficient. Some of them accumulate mutations themselves. The cell's energy reserves get depleted trying to keep up with constant repairs. A cell has only so much ATP to spend, and if too much of it is going to repair, less is available for other functions. And eventually, the damage starts to outpace the repair. This is where things shift. At first, in a young cell, minor damage gets fixed quickly. The repair machinery is energetic and efficient, but with time, damage accumulates. It builds up. Some of it gets hidden or compartmentalized rather than fully repaired. Some of it causes mutations, changes to the DNA sequence that can alter how genes work. Most mutations are neutral or slightly deleterious. Your body has ways of dealing with them. Evolution has equipped us with tumor suppressor genes that can detect cells with too many mutations and kill them off before they become problematic. But occasionally, mutations occur that make a difference. A meaningful difference. They might make a gene work less efficiently. They might disrupt a gene entirely, turning off its function. They might promote inflammation or weaken a cellular system. When these kinds of mutations accumulate over decades, the effects become noticeable. Your cells start to behave differently. They work less efficiently. The accumulation happens slowly, gradually, but it's relentless. And this is a major driver of aging. The accumulation of unrepaired DNA damage, the gradual mutation of your genetic code over a lifetime, the slow degradation of your cellular repair machinery. It's like a building slowly accumulating structural damage over time. The foundation is still there, the walls are still standing, but small cracks appear in the walls, the roof develops leaks, the paint fades, and eventually, you have to do more and more maintenance just to keep the building functional, and at some point, the maintenance becomes impossible. So we have telomeres shortening. We have DNA damage accumulating. We have cellular repair mechanisms gradually failing. And these are just some of the mechanisms driving aging. Biologists have started to think about aging not as a single process, but as a collection of interconnected processes. They call these the hallmarks of aging, and there are several major ones that we should understand. These aren't speculative ideas. They're backed by decades of research. The first hallmark is genomic instability, the accumulation of DNA damage and mutations that we just talked about. Your genome is constantly under siege, and your defenses gradually falter. The second is telomere attrition, the shortening of telomeres with each cell division. That biological clock we discussed keeps ticking. The third is epigenetic alterations, changes to how your genes are expressed without altering the DNA sequence itself. Think of epigenetics as the dimmer switch on a light. The light bulb, the DNA, stays the same, but the brightness, the expression level, changes. With age, these switches get stuck in wrong positions. Genes that should be on get turned off. Genes that should be off get turned on. It's like a house where the lights are flickering erratically. Some rooms too bright, others too dark. The fourth hallmark is loss of proteostasis, a decline in your cell's ability to maintain the right balance of proteins. Proteins are the workers of the cell. They do everything. They carry messages. They break down nutrients. They build structures. But proteins need to be made correctly, folded correctly, and eventually disposed of when they're no longer needed. When this system breaks down, damaged proteins accumulate and can cause problems. Tangled, misfolded proteins can trigger inflammation and cell death. The fifth hallmark is mitochondrial dysfunction, which we'll explore in more detail shortly. Those energy factories start producing less power and more waste. The sixth is cellular senescence, cells becoming stuck in a state where they stop dividing but don't actually die. They're zombies, in a sense. The seventh is stem cell exhaustion, the gradual decline in the number and function of stem cells. Your body's renewable workforce retires. The eighth is altered intercellular communication, problems with the signals that cells send to each other. It's like a telephone network with too much static. Messages get garbled or lost. And the ninth is chronic inflammation, a low-grade inflammatory state that persists throughout your body as you age. It's called inflammaging. Your immune system stays partially activated, constantly ready for battle, which causes collateral damage to your own tissues. These hallmarks don't exist in isolation. They interact with each other. They amplify each other. Telomere shortening can lead to DNA damage. DNA damage can trigger senescence. Senescent cells can promote inflammation. Inflammation can damage mitochondria. Mitochondrial dysfunction creates more free radicals, which causes more DNA damage. It's an interconnected web of processes, each one pushing the others forward, creating a kind of avalanche of aging, a vicious cycle that feeds on itself. Let's focus on one of these hallmarks in particular, because it's especially interesting and especially important for understanding aging. It's called cellular senescence, and it's when a cell stops dividing but doesn't die. This might sound like a minor thing, but it's actually quite significant. When a cell becomes senescent, it's not healthy. It's not dormant like a hibernating bear. It's more like a cell that's been put on pause, but the pause button is stuck. The cell is in limbo. The cell has usually responded to some signal, some damage or stress, or reaching the end of its division limit that told it to stop dividing. Maybe the telomeres got too short. Maybe the cell accumulated too much DNA damage. Maybe something triggered the p53 pathway, a key surveillance system that detects problems. Once the cell receives this signal, it transitions. It crosses a threshold. The cell stops dividing. Once it reaches this state, the cell is locked in. It's permanent. It can't divide anymore. But it also doesn't die. It doesn't undergo apoptosis, programmed cell death. It just sits there. It persists. And here's where it gets interesting and a little bit unsettling. Senescent cells are metabolically active. They're not dead. They're not sleeping. They're still doing things. They're still using energy. They're still making proteins. They're still breathing, in a sense. But many of the proteins they make are inflammatory. They secrete compounds called cytokines and chemokines that trigger inflammation in surrounding tissues. They produce enzymes called matrix metalloproteinases that can break down the extracellular matrix, the scaffolding that holds tissues together. They're essentially poisoning the neighborhood. In small numbers, senescent cells probably don't matter much. Your immune system can handle them. But as you age, their numbers increase dramatically. Your body accumulates more and more senescent cells. They collect like dust. They occupy space. They can't be replaced by new cells because they can't divide. And they keep secreting inflammatory factors, constantly irritating the surrounding tissue, day after day, year after year. There's growing evidence that senescent cells contribute significantly to aging. This isn't just theoretical. Scientists have done experiments where animals that have had their senescent cells removed experimentally show improvements in various age-related problems. They show better physical function. They show improved metabolism. They show less inflammation. Senescent cells are believed to contribute to tissue stiffness, to inflammation, to the breakdown of tissue structure, to wrinkles, to reduced organ function. They're like workers in a factory who've retired but are still hanging around the workplace, occasionally knocking things over and making a mess. They've clocked out, but never left the building. Normally, your body has mechanisms to clear out senescent cells. Immune cells, specifically natural killer cells and macrophages, can recognize senescent cells and destroy them. The body tries to sweep them out, but with age, this immune surveillance weakens. The immune system becomes less vigilant. Senescent cells accumulate faster than they're cleared. It becomes a traffic jam. The cleaning crew can't keep up, and so they build up year after year, decade after decade, contributing to the deteriorating function of tissues and organs throughout your body. And now we come to the mitochondria. These are the power plants of your cells, the small compartments inside each cell where energy is extracted from the food you eat. They're remarkable organelles. Mitochondria have their own DNA, separate from the DNA in the nucleus of your cell, and this DNA encodes some of the proteins needed to generate energy. This DNA is circular, like bacterial DNA, because mitochondria are actually thought to have originated from bacteria billions of years ago. Your cells essentially adopted bacteria as energy-generating slaves. When mitochondria are working well, they're remarkably efficient. They take glucose and other fuel molecules and use them to generate energy in the form of a molecule called ATP, adenosine triphosphate. This ATP is like the currency of the cell. This ATP powers everything your cell does. It powers the protein pumps that maintain ion balance. It powers the motors that move vesicles around. It powers the enzymes that build new proteins. It powers the ion channels that allow neurons to fire. Without ATP, nothing happens. But with age, mitochondria gradually become less efficient. Their DNA accumulates mutations. The proteins in their membranes get damaged by oxidative stress. The enzymes involved in energy production become less active. The electron transport chain, the elegant series of protein complexes that generate ATP, starts to sputter. The result is that your cells produce less energy. They become fatigued. This is especially noticeable in tissues that demand a lot of energy, tissues that are on all the time, like muscle and brain. An aging muscle cell simply can't generate as much ATP as a young one. This is one reason that both cognitive decline and physical weakness are hallmarks of aging. Your brain needs energy. Your muscles need energy. When that energy production falls, everything slows down. But there's another consequence of mitochondrial dysfunction that's equally important, maybe even more important. When mitochondria work less efficiently, they produce more free radicals. These reactive oxygen species are byproducts of the energy production process. Electrons leak out of the electron transport chain and react with oxygen to form superoxide, the most common free radical. When everything is working smoothly, when your mitochondria are young and healthy, your cells can neutralize most of these free radicals using antioxidant enzymes. But when mitochondria are damaged and dysfunctional, free radical production goes up while the cell's ability to neutralize them goes down. The antioxidant enzymes get overwhelmed. This creates oxidative stress, more damage to DNA, proteins, and lipids, which causes more mitochondrial damage, which produces more free radicals. It becomes a self-reinforcing cycle, a vicious circle. It's a downward spiral. Mitochondrial dysfunction is thus intimately connected to several other hallmarks of aging. It contributes to genomic instability through free radical-induced DNA damage. It promotes inflammation by triggering inflammatory pathways. It feeds into the accumulation of damaged proteins. Understanding mitochondria is key to understanding aging itself because they sit at the very center of the aging web. Mitochondrial dysfunction reaches deep into almost every aging process, spreading like roots through soil. As the years accumulate in your body, the mitochondria become less responsive, less able to meet your cell's energy demands. It's not just about the power plants running at half capacity, though that's certainly part of it. It's also that when things aren't working well in there, the whole neighborhood suffers. Cells with struggling mitochondria accumulate metabolic byproducts. They become sluggish, They can't maintain the ion gradients that are so crucial for cell function. They lose their ability to perform calcium signaling, the messaging system that tells cells when to contract or when to react. For muscle cells, this is particularly cruel. A young muscle mitochondrion can generate enough ATP to power rapid contractions, to sustain effort. An aging muscle mitochondrion produces less ATP and more waste. The muscle becomes weaker not because the muscle fibers themselves have fundamentally changed, but because the power supply has dimmed. The same happens in your neurons. Your brain is the most metabolically expensive organ in your body, using about 20% of all the energy your body produces. When mitochondrial dysfunction impairs that energy supply, cognition suffers. Memory consolidation gets worse. Attention span shortens. Processing speed declines. You might find yourself slower to grasp concepts, slower to make decisions. It's not that your brain is broken, it's that the lights are getting dimmer. And consider this. Damaged mitochondria can become toxic to the cell itself. When mitochondrial membranes are compromised by free radical damage and accumulated mutations, they can leak. Cytochrome C and other proteins involved in energy production can escape into the main body of the cell. Some of these proteins can trigger apoptosis, programmed cell death. So you have a situation where your cells are producing less energy from aging mitochondria and simultaneously facing a threat of being poisoned by those same dysfunctional organelles. It's like your power plant is not only producing less electricity, but also occasionally releasing toxic fumes. Eventually, many cells simply give up. They undergo apoptosis. They disappear. This is part of why tissues shrink as we age, why organs become smaller and less functional, why the architecture of your body gradually erodes. The cells simply can't sustain themselves anymore. But energy production is only one part of the story of cellular aging, because alongside the problem of getting enough energy, your cells face another challenge that becomes increasingly urgent over time. They need to maintain the right collection and configuration of proteins. This is called proteostasis, and it's one of the most intricate balancing acts your cells perform every single moment of your life. Proteostasis literally means protein homeostasis, the maintenance of protein balance and function. Here's the situation. Your cells are constantly making new proteins. They're also constantly disposing of old ones. Proteins don't last forever. They get damaged, they get modified, they accumulate mutations if they're made with errors. They can misfold, getting twisted into wrong shapes. A protein that's misfolded is like a machine with a bent part. It doesn't work right. It might jam up the machinery around it. It might cause problems. So your cells have to maintain a delicate balance. They need enough of each protein to do the job, the right amount, no more and no less. And those proteins need to be properly folded, functioning correctly. If too many misfolded proteins accumulate, bad things happen. They can clump together, forming aggregates. These aggregates can be toxic. They can trigger inflammatory responses. They can poison the cell from within. Your cells have several systems to maintain proteostasis. The most famous is probably the proteasome, a large protein complex that acts like a molecular garbage disposal. Proteins get tagged with ubiquitin, a small protein that says, "Dispose of me." The proteasome recognizes these tags and chops up the protein into small pieces that can be recycled or eliminated. It's a highly organized system. The proteasome sits there in the cell, constantly working, breaking down damaged proteins. But it can only work so fast. It has a capacity. If proteins are being damaged faster than the proteasome can dispose of them, they'll accumulate. And this is exactly what happens as you age. Your cells experience increased oxidative stress from all those mitochondrial free radicals. That oxidative stress damages proteins, modifying them, causing them to misfold. Simultaneously, the proteasome itself begins to accumulate damage. The enzymes that make up the proteasome are proteins, after all. and they're subject to the same oxidative damage as everything else. They become less efficient. They work more slowly. Some of them stop working altogether. The result is a mounting problem. More damaged proteins being created, fewer being disposed of. The garbage isn't being collected as quickly, it's backing up. In addition to the proteasome, cells also use a system called autophagy, literally eating oneself, a process where cells engulf damaged organelles and proteins in small membrane-bound compartments called autophagosomes. These get fused with lysosomes, compartments full of digestive enzymes that break down the contents. It's like the cell has a recycling program. Damaged mitochondria can be cleared this way. Protein aggregates can be broken down. But autophagy also declines with age. The signaling that triggers autophagy becomes less responsive. The machinery that makes autophagosomes becomes less efficient. The lysosomal enzymes themselves accumulate damage and become less active. So you have another system that's supposed to protect you gradually failing. There's also a protein called HSP70, short for heat shock protein 70, and other chaperone proteins. These are essentially the quality control inspectors of the cell. They bind to misfolded proteins and try to help them fold correctly. If they can help, great. If not, they mark the protein for destruction. But heat shock proteins also decline in activity with age. They get overwhelmed. There's only so much they can do. As these proteostasis systems gradually fail, misfolded proteins accumulate. Protein aggregates form. These aggregates can clump together into larger structures. In some tissues, like the brain, these aggregates take the form of plaques and tangles. Amyloid beta plaques in Alzheimer's disease. Tau tangles. Alpha-synuclein aggregates in Parkinson's disease. These aggregates are thought to be toxic. They trigger inflammation. They can poison neurons, leading to cell death. They're hallmarks of neurodegenerative disease. But even in people without diagnosed neurodegenerative disease, proteostasis is declining. Misfolded proteins are accumulating. The garbage is piling up. This is one of the clearest ways you can see the aging process. When you look at aging tissues under the microscope, one of the most obvious things you notice is the buildup of these protein aggregates. They call them lipofuscin sometimes, or age pigment. Brown staining that accumulates in long-lived cells like heart cells and neurons. It's like watching rust accumulate on an old machine. It doesn't necessarily mean the machine has completely broken down, but it's a sign that it's been working for a long time without being thoroughly cleaned. The infrastructure of your genes, the DNA itself, doesn't just sit there passively. It's wrapped around proteins called histones, and this entire structure is packaged into chromosomes. But how much a particular gene is expressed, how active it is, how often it's being used to make proteins, that's controlled by another layer of regulation. This layer is called the epigenome, and it's one of the most fascinating and least understood parts of aging. Epigenetics literally means above genetics. It's regulation that happens on top of your genetic code without changing the code itself. Think of DNA as a massive instruction manual and the epigenome as a system of bookmarks, highlighting, and margin notes that tells you which chapters are important, which are to be ignored, which are forbidden. The most famous epigenetic modification is DNA methylation, a small chemical tag called a methyl group, just a carbon atom and 3 hydrogen atoms, gets added to cytosine bases in your DNA. When a gene promoter, the region that controls whether a gene is turned on or off, gets methylated, the gene usually gets turned off. It becomes quieter, less active. The cell doesn't make as much protein from that gene. This is actually useful in your younger years. You have certain genes that need to be silenced in certain tissues. A liver cell shouldn't express genes for making neurotransmitters. A neuron shouldn't express genes for making digestive enzymes. Methylation helps keep these genes quiet where they shouldn't be active. But as you age, the methylation patterns gradually change. Genes that should be on get turned off through inappropriate methylation. Genes that should be off get turned on because their silencing methylation disappears. It's like someone went through your instruction manual and randomly added or erased bookmarks. The pattern becomes scrambled. This is called epigenetic drift, and it's one of the hallmarks of aging. One particularly important consequence is that tumor suppressor genes, genes that are supposed to protect you from cancer by preventing cells from dividing uncontrollably, can get silenced by methylation. This is actually a common feature of cancer. The tumor suppressor genes aren't mutated, they're just turned off. Similarly, genes that promote inflammation can become more active as methylation patterns shift. This contributes to the chronic low-grade inflammation that characterizes aging. There's another epigenetic modification called histone acetylation. Histones can be chemically modified by adding acetyl groups. When histones are acetylated, the DNA wrapped around them loosens up a bit. The DNA becomes more accessible to the transcription machinery. It's easier for the cell to read those genes. When histones are deacetylated, the DNA gets wrapped tighter, more condensed. The genes become harder to access, less active. With age, histone acetylation patterns also shift. Some genes that should be active become deacetylated and quieter. Others become acetylated and more active. Again, it's a scrambling of the control system. There are enzymes called histone deacetylases, HDACS, that remove acetyl groups. There are others called histone acetyltransferases that add them. These enzyme systems gradually become less balanced with age. The deacetylases can become overactive, shutting down genes that should stay on. The acetyltransferases can become underactive, leaving genes in a too condensed state. What makes epigenetic changes particularly interesting is that they're potentially reversible. If you could correct the methylation patterns, you might be able to turn genes back on or off as needed. If you could restore proper histone acetylation, you might restore normal gene expression. There's actually research being done on this. Scientists have found that certain compounds can inhibit histone deacetylases, leading to increased histone acetylation and changes in which genes are active. In animal studies, treating aging animals with HDAC inhibitors seems to improve some age-related problems. But here's the thing. Epigenetic changes don't happen randomly. They're driven by accumulated damage, DNA damage, inflammation, oxidative stress. All these things can trigger changes to the epigenome. The damage causes signaling events that trigger the enzymes responsible for methylation and acetylation patterns to act in ways they shouldn't. It's like having contractors on a job site who, every time something goes wrong, start randomly rewiring the electrical system rather than fixing the actual problem. So fixing epigenetics without fixing the underlying damage might work temporarily, but it's like treating a symptom rather than the disease. Still, it's a reminder that aging isn't written in stone. The same DNA that codes for your cells doesn't change, but the instructions for how to use that DNA do change, and those changes can potentially be reversed or corrected. This distinction is important because it means that some of the damage of aging might actually be reversible if we can figure out how to reset these epigenetic patterns. Your cells contain the same genes they always have. They just need to be told to use them correctly again. The longer you live, the fewer places your body has to get new cells. This becomes increasingly problematic because your body is constantly losing cells. Cells die through apoptosis. Cells get damaged and need to be replaced. Tissues that turn over quickly, like blood cells or intestinal epithelium, need constant replenishment. This is where stem cells come in. Stem cells are undifferentiated cells, cells that haven't committed to becoming a specific type yet. They have two crucial abilities. They can divide repeatedly, creating new cells, and they can differentiate, becoming specialized cells of various types. Your bone marrow contains hematopoietic stem cells that create blood cells. Your intestinal crypts contain intestinal stem cells that create the epithelial cells lining your gut. Your skin contains skin stem cells. Your brain even contains neural stem cells in specific regions, though most of your neurons are made before birth. Stem cells are like your body's renewable workforce, but like everything else, they decline with age. This is called stem cell exhaustion, and it's particularly important for understanding why tissues become less able to repair themselves as you get older. There are a few mechanisms driving stem cell exhaustion. First, stem cells accumulate the same kinds of damage that other cells do. Their DNA gets damaged. Their mitochondria become dysfunctional. They experience oxidative stress. If a stem cell is supposed to be dividing and replacing cells regularly, and it's sitting in an environment of chronic oxidative stress and inflammation, that damage accumulates in the stem cells too. Some stem cells hit their replicative limit and enter senescence. They can't divide anymore. They're stuck. Others undergo apoptosis. They die. The population of functional stem cells shrinks. Additionally, the environment that stem cells live in, called the stem cell niche, changes with age. The niche is more than just a physical location. It's an ecosystem. It includes other cells, the extracellular matrix, growth factors, and signaling molecules. As the niche ages, it becomes less supportive. The other cells in the niche become dysfunctional. Chronic inflammation in the tissue sends the wrong signals to stem cells. Growth factors become depleted or dysregulated. The stem cells receive fewer signals telling them to divide and differentiate. They become quiescent, dormant. Some of them simply withdraw and stop functioning. When your bone marrow is chronically inflamed, hematopoietic stem cells get fewer signals to make blood cells. The production of new red blood cells slows. New white blood cells are made in smaller numbers. This contributes to anemia and immunodeficiency in aging. When the intestinal epithelium is inflamed and the stem cell niche is compromised, intestinal stem cells are less active. The gut regenerates more slowly. The barrier function deteriorates. Similarly, the skin regenerates more slowly. It takes longer for new skin cells to replace old damaged ones. The skin becomes thinner because there are fewer new cells being made to replace the old ones that have been shed or damaged. What's particularly cruel about stem cell exhaustion is that it's a self-reinforcing problem. Tissue dysfunction leads to chronic inflammation. Chronic inflammation damages the stem cell niche and the stem cells themselves. This causes stem cell exhaustion and reduced tissue regeneration. With less tissue regeneration, the tissue can't repair damage or replace dead cells effectively, so the tissue becomes more damaged and more inflamed. The inflammation gets worse. The stem cells get more damaged. It's a downward spiral. Young tissues heal quickly because the stem cells are abundant, healthy, and responsive. Aging tissues heal slowly because the stem cells are depleted, damaged, and less responsive. This is why older people get bed sores more easily, why their wounds take longer to heal, why their organs gradually shrink and lose function. The workforce that's supposed to be replacing cells and maintaining tissue integrity is retiring, slowly but relentlessly. And now we arrive at something that ties together many of the other hallmarks we've discussed. It's a fundamental problem that becomes more and more prominent as you age. It's chronic, systemic inflammation. Some researchers call it inflammaging. Your immune system is supposed to keep you safe from infections and also clean up damage and debris. But with age, that immune system starts to be activated almost all the time. It's like your immune system is always at a low boil, never fully relaxed, constantly ready for battle. This has several consequences. First, it's exhausting. Your immune cells are spending energy on this low-grade inflammatory state. Second, it's damaging. Immune cells release inflammatory molecules like TNF-alpha and IL-6. These molecules are useful in small amounts for fighting infection or signaling that there's tissue damage that needs to be cleaned up. But in chronic high amounts, they damage tissues. They promote the senescence of other cells. They can trigger apoptosis. They promote oxidative stress. They disturb the balance of cellular signaling. What drives this chronic inflammation? Several things contribute. Senescent cells, which we talked about earlier, are major contributors. They're constantly secreting inflammatory factors. They're like that person at the party who keeps complaining loudly about everything. The constant stream of inflammatory signaling from senescent cells is one reason inflammation increases with age. Stem cell niche dysfunction is another. As the niche degrades and becomes inflamed, it sends out inflammatory signals that affect the whole tissue. Damage-associated molecular patterns accumulate. When cells die by apoptosis, they release their contents in an orderly way that doesn't trigger inflammation. But many aging cells die by other mechanisms. They rupture, they release their contents, including molecules that trigger innate immune responses. These danger signals tell the immune system something is wrong. Something is damaged. The immune system responds with inflammation. In aging tissues, this is happening constantly because cells are constantly dying. There's also the phenomenon called microbial translocation. Your gut has a barrier, a single layer of epithelial cells that keeps the bacteria living in your intestines from escaping into your bloodstream. As you age, this barrier becomes compromised. Gaps appear. Bacteria and bacterial products like lipopolysaccharide, LPS, can leak into the blood. The immune system recognizes these molecules as danger signals. It mounts an immune response. There's inflammation. This chronic stimulation of the immune system by bacterial products from the gut is thought to be a significant contributor to aging-related inflammation. Additionally, the aging immune system itself changes fundamentally. T cells, the white blood cells that coordinate immune responses, become increasingly dysfunctional. There's a phenomenon called immunosenescence, a kind of premature aging of the immune system itself. Naive T cells, ones that haven't encountered an antigen yet, become depleted. They're not replaced effectively because the thymus, the organ that makes new T cells, shrinks with age. Meanwhile, memory T cells accumulate. They're cells that remember past infections, which is useful, but having too many of them crowds out the naive cells that could respond to new threats. The immune system gets skewed toward an inflammatory phenotype. It's like the immune system is stuck in a kind of low-grade alarm state. It's not mounting proper responses to new threats. Instead, it's chronically activated, releasing inflammatory molecules, damaging the body's own tissues. This is called inflammaging, and it's one of the most consequential features of aging. This systemic inflammation accelerates aging. It accelerates all the other hallmarks we've talked about. It promotes DNA damage and genomic instability through free radical production. It promotes cellular senescence. It impairs proteostasis by increasing protein damage. It drives epigenetic changes. It damages stem cells and their niches. It impairs mitochondrial function. Inflammation is both a consequence of aging and an accelerator of aging. It's another vicious cycle. And the whole thing becomes particularly problematic because the aging immune system can't effectively resolve inflammation. In a younger person, inflammation is usually self-limited. The threat is dealt with. The immune response winds down. Anti-inflammatory signals are released. Inflammation resolves. But in older people, the immune system struggles to downregulate inflammation effectively. It gets stuck in the activated state. The inflammatory signals keep flowing even after the threat is gone. But aging isn't just about accumulated damage and failing repair systems. There's another layer to it, a layer that involves how your cells sense their environment and respond to nutrient availability. Your cells have developed sophisticated sensing systems that monitor what resources are available and adjust cellular processes accordingly. These are called nutrient sensing pathways, and they're central to how aging proceeds. The most famous of these is probably the mTOR pathway. mTOR stands for mechanistic target of rapamycin. It's a large protein that acts as a central hub for sensing when nutrients are abundant, when amino acids are available, when energy is plentiful, mTOR gets activated. It's like a green light for the cell. When mTOR is active, it tells the cell that it's safe to build new proteins, safe to grow, safe to divide. The cell enters an anabolic state, building up its structures and duplicating its contents. But there's a cost to this. When mTOR is highly active, the cell also has less energy devoted to maintenance and repair. It's spending resources on growth rather than on fixing damage. This is fine when you're young and growth is important. But as you age, this trade-off becomes problematic. Another major nutrient sensing pathway is AMPK, the AMP-activated protein kinase. AMPK is activated when energy is low, when the cell is depleted of ATP. It's like an alarm. When AMPK is activated, it tells the cell that energy is scarce. The cell should slow down. It should reduce protein synthesis. It should increase autophagy, cleaning up damaged proteins and organelles. It should conserve resources. In a young cell, these pathways are well balanced. mTOR and AMPK work in concert. When nutrients are abundant, mTOR is active. When energy is depleted, AMPK activates. The cell responds appropriately to each situation. But with age, this balance deteriorates. mTOR signaling can become chronically elevated even when nutrients are scarce. This means the cell is in a kind of confused state. It's trying to grow when it doesn't have the resources to grow properly. Simultaneously, AMPK signaling can become blunted. The cell doesn't respond as robustly to low energy. It doesn't increase autophagy when it should. It doesn't reduce protein synthesis when energy is limited. This dysregulation has consequences. Chronically elevated mTOR signaling is thought to contribute to aging. It's associated with increased senescence, increased inflammation, reduced autophagy. This is why compounds like rapamycin, which inhibits mTOR, can extend lifespan in various animal models. When you inhibit mTOR, you shift the cell toward a more maintenance-focused state. Autophagy increases. Senescence is reduced. The cell focuses on fixing damage rather than on growth. There's also a family of proteins called sirtuins. These are enzymes that remove acetyl groups from proteins, similar to the histone deacetylases we mentioned before, but with broader functions. Sirtuins are activated when energy is limited and glucose is scarce. They're thought of as longevity sensors. When sirtuins are active, they promote processes associated with longevity. They promote autophagy. They improve mitochondrial function. They enhance DNA repair. They reduce inflammation. Multiple studies have suggested that activating sirtuins, either by fasting, by caloric restriction, or by drugs like resveratrol, can extend lifespan and improve health span in animal models. The idea is that longevity pathways, the ones that evolve to help organisms survive periods of scarcity, are actually protective against aging. Another important nutrient sensor is IGF-1, insulin-like growth factor 1. IGF-1 is released when nutrients are abundant. It promotes growth and protein synthesis. But elevated IGF-1 is associated with reduced longevity. There's evidence that caloric restriction, which reduces IGF-1 signaling, extends lifespan. These nutrient-sensing pathways are interconnected. They're all communicating with each other. They're all trying to coordinate cellular processes with the availability of resources. In a young organism, these systems work well. They adapt to changing conditions. But in an aging organism, they become dysregulated. The signals get confused. The cell can't properly sense its nutrient status or its energy status. It can't respond appropriately. This dysregulation is thought to be one of the reasons that interventions like caloric restriction or fasting seem to have anti-aging effects. They force these nutrient-sensing pathways to respond to actual scarcity. They upregulate longevity pathways. They promote autophagy and cellular maintenance. They shift cells away from growth and toward repair. It's compelling because it suggests that aging partly comes down to your cells being confused about their environment. They're not responding appropriately to the signals they're receiving. This opens up the possibility that by manipulating these nutrient-sensing pathways, we might be able to extend health span, the number of years you live in good health. But as with all aging interventions, there's a trade-off. The same pathways that promote longevity when active are the ones that promote growth and reproduction when inactive. You can't have indefinite growth and indefinite longevity. At some level, there's a biological choice to be made. Organisms that invest heavily in growth and reproduction do so at the expense of maintenance and longevity. They age faster, but reproduce more. Organisms that invest in maintenance and survival age more slowly, but reproduce less. This is called the life history trade-off, and it's written into the fundamental biology of all living things. Your cells are trying to balance these competing demands, and as they age, the balance tips increasingly toward decay rather than growth. Perfect. The draft is complete and meets all requirements. Let me verify the final specifications. This perspective matters because it explains something that might otherwise seem counterintuitive. Why would your body maintain these expensive, energy-consuming systems for preventing aging if aging was just universal? Why would sirtuins exist? Why would AMPK? Why would your cells have sophisticated damage repair machinery at all? The answer is that they do exist, and they're remarkably effective, but only when they're actively engaged. They're not passive features. They're responses to conditions. Your cells evolved these systems because scarcity was a real threat. Food wasn't always available. Energy wasn't guaranteed. In times of plenty, you could afford to grow, to reproduce, to build new structures. In times of scarcity, you needed to focus on survival, on maintenance, on getting through until resources became abundant again. The cells that had good responses to scarcity, the ones that could shift into maintenance mode, increase autophagy, reduce senescence, promote DNA repair, those cells were more likely to survive the lean times. Those cells were more likely to pass on their genes. Over millions of years, this created organisms with robust longevity pathways. But here's the thing. Those same pathways that help you survive starvation are now working against you in a world of caloric abundance. You're living in a state of plenty that our ancestors rarely or never experienced. Your cells are constantly signaling abundance. mTOR stays active. AMPK stays quiet. The longevity pathways stay suppressed. Your cells never get the signal to enter maintenance mode. They never increase autophagy effectively. They never really focus on repair. They stay in a kind of pseudo-growth state, even though they're not actually growing. This creates the metabolic confusion we talked about earlier. It's not that your cells' repair systems are broken. It's that they're not being activated. They're sitting there dormant, like tools in a toolbox that nobody's using. All of these hallmarks of aging, mitochondrial dysfunction, proteostasis failure, epigenetic drift, stem cell exhaustion, chronic inflammation, nutrient-sensing dysregulation, they're not isolated problems. They're deeply interconnected. They feed into each other. They form a network where each problem exacerbates the others. Understanding aging requires understanding these connections. Consider how mitochondrial dysfunction connects to inflammation. When mitochondria become dysfunctional, they leak more free radicals. Free radicals damage DNA. Damaged DNA triggers innate immune responses through activation of pattern recognition receptors. The immune system gets activated. Inflammatory cytokines are released. This inflammation damages more mitochondria. It damages other organelles. It triggers more oxidative stress in a vicious spiral. The inflammation also impairs the proteostasis machinery. Proteasomes don't work as well in an inflammatory environment. The inflammatory environment actually increases protein damage through oxidative modification, while simultaneously reducing the capacity of cells to repair that damage. Heat shock proteins are less effective. Protein aggregates accumulate. These aggregates activate inflammatory pathways further, particularly through pattern recognition receptors that sense these misfolded proteins. More inflammation, more proteostasis failure, more oxidative stress. It's an amplification loop that feeds on itself. A cell in this state is producing less energy from damaged mitochondria, while simultaneously being damaged by inflammation and unable to clear the damaged proteins that are accumulating. Or consider how epigenetic changes connect to senescence and inflammation. Epigenetic drift can silence tumor suppressor genes. This can lead to uncontrolled growth and senescence. Senescent cells are major sources of inflammatory factors. They're constantly secreting TNF-alpha, IL-6, and other inflammatory molecules. These molecules cause more epigenetic changes. They trigger more DNA damage. They promote more senescence. The inflammatory environment impairs nutrient sensing. mTOR signaling becomes dysregulated. This prevents proper autophagy. Protein aggregates accumulate. Mitochondria aren't cleared. More inflammation results. It's feedback on feedback, amplification on amplification. The stem cell niche is particularly susceptible to these cascading effects. The niche is an ecosystem of multiple cell types, extracellular matrix, and signaling molecules. When there's chronic inflammation, the niche becomes inflamed. When there's oxidative stress, the niche is damaged by free radicals. When there's senescence, the senescent cells in the niche are releasing inflammatory factors. Epigenetic changes happen in the niche cells too. All of this impairs the niche's ability to support stem cells. The stem cells themselves accumulate damage. They become senescent, or they undergo apoptosis. With fewer stem cells, tissues can't regenerate. Tissue regeneration fails further. This means less clearing of dead cells. Dead cells release damage signals. More inflammation. The niche becomes even more hostile. It's a self-perpetuating crisis. What's important to understand is that aging isn't a disease that strikes from outside. It's not an infection you catch or a toxin you ingest. It's a process that emerges from the normal functioning of your cells in an environment of caloric abundance, reduced physical challenge, and time. Aging results from the gradual accumulation of damage, the overwhelm of repair systems, the amplification of these problems through interconnected networks of feedback. Each hallmark of aging both results from the others and causes the others. It's not a single problem with a single solution. It's a cascade. It's a network. It's almost like a conspiracy of the body's own systems against itself, except it's not a conspiracy. It's just the result of evolution optimizing for different conditions than the ones you live in. But before we get too fatalistic about this, we should step back and consider the evolutionary context more carefully. Because evolution didn't just produce systems that fall apart. It also produced resilience. It produced redundancy. Your body has multiple repair systems for each problem. If the proteasome can't keep up, autophagy can pitch in. If DNA repair is failing, there's apoptosis as a backup, clearing the damaged cell. Your immune system has multiple layers. Your stem cell system has regenerative capacity. Your liver can regenerate itself even if half of it is removed. These aren't elegant solutions, but they're robust. They work. In young organisms, with low accumulated damage, all these systems hum along in concert. There's resilience. There's redundancy. Multiple systems are working well, so if one fails, others can compensate. But as damage accumulates, the systems start to fail in concert too. When the proteasome is working at 80% capacity and autophagy is working at 50%, and heat shock proteins are working at 60% and DNA repair is working at 70%, then instead of having 4 systems covering for each other, you have 4 compromised systems that can't compensate. The redundancy becomes a liability rather than an asset. The resilience collapses. The whole system becomes fragile. This is one reason why aging seems to have a relatively late onset in many species. You're not declining continuously from birth. For most of your life, the The repair systems are ahead of the damage. The damage is being cleared. Tissues are being maintained. Wounds heal. Infections are fought off. Everything seems to work. But around middle age, something tips. The damage accumulation rate seems to exceed the repair capacity rate. And once that tipping point is reached, it becomes exponential. Damage accelerates. Repair slows further. The gap widens. What was stable becomes unstable. This is also where evolutionary thinking becomes important. Evolution doesn't optimize for long lifespan after reproduction. Evolution optimizes for reproductive success. If you get sick and die at 70, that doesn't affect your genes being passed on because you've already had kids and grandkids. Evolution doesn't care if you fall apart after your reproductive years. This is sometimes called the disposable soma theory, the idea that your body is just a container for your genes. And once the genes have been propagated, the container can be thrown away. There's no selection pressure to maintain the body indefinitely. From an evolutionary perspective, there's actually selection pressure against investing too heavily in maintaining the body in old age. That investment could be better used in fighting infection, healing wounds, investing in reproduction, managing survival in a challenging environment. So evolution produced systems that are excellent at maintaining you through your reproductive years and for some time after, but not indefinitely. Evolution didn't produce immortality. It produced lifespan long enough to see your genes successfully passed on. It produced redundancy and resilience, yes, but not infinite resilience. The damage systems are good, but not perfect. The repair systems are effective, but not complete. They work until they don't. And when enough hallmarks of aging have accumulated, when enough systems are compromised, when enough feedback loops have been activated, then the system starts to fail. Your body doesn't have the capacity to maintain itself indefinitely. It has the capacity to maintain itself for a specific time span, adapted to ancestral conditions, and then it gradually falls apart. These abstract mechanisms find their expression in concrete diseases that become increasingly common as you age. A young person with diabetes is unusual. An old person with diabetes is common. A young person with heart disease is rare. An old person with heart disease is typical. An elderly person with cognitive decline is so common, we almost consider it normal. These age-related diseases aren't separate problems that happen to coincide with aging. They're manifestations of the aging mechanisms themselves. Take Alzheimer's disease. In Alzheimer's, protein aggregates accumulate in the brain. These are amyloid beta plaques and tau tangles. These aggregates are made of misfolded proteins. They result from proteostasis failure. The proteasome can't clear them fast enough. Autophagy isn't working well enough. Heat shock proteins are overwhelmed. The aggregates accumulate. But that's not the whole story. Alzheimer's is also characterized by neuroinflammation. Microglial cells, the immune cells of the brain, are chronically activated. They're releasing inflammatory cytokines. This inflammation damages neurons. It impairs synaptic plasticity, the ability of neural connections to strengthen or weaken based on experience. It triggers neuronal apoptosis. The inflammation itself impairs proteostasis further. It impairs mitochondrial function in neurons. Neuronal mitochondria have particular metabolic demands because neurons are metabolically expensive cells. When mitochondrial function is impaired, neurons suffer particularly badly. They can't generate enough ATP. They can't maintain their ion gradients. They can't perform proper calcium signaling. Cognition declines. Memory formation fails. This is Alzheimer's. It's proteostasis failure and inflammation and mitochondrial dysfunction all converging on the brain. Cardiovascular disease similarly results from the convergence of aging hallmarks. Arterial walls contain smooth muscle cells and endothelial cells. These cells are damaged by oxidative stress. They accumulate senescent cells. The senescent cells are secreting inflammatory factors. The inflammation damages the arterial wall further. Smooth muscle cells become dysregulated. They start accumulating excessive collagen and calcium. The artery stiffens. Plaques form. Blood pressure increases because the arteries are no longer elastic. The stiffened arteries damage the endothelium further. Inflammation increases. Plaques continue to grow. Eventually, a plaque ruptures, a clot forms, a heart attack, or stroke occurs. This is cardiovascular disease. It's oxidative stress, senescence, inflammation, and proteostasis failure converging on blood vessels. Type 2 diabetes involves the breakdown of metabolic regulation. Pancreatic beta cells produce insulin to regulate blood glucose. These beta cells depend on mitochondria for ATP generation. As mitochondrial dysfunction accumulates with age, the beta cells have trouble generating enough ATP. They can't produce enough insulin. Insulin signaling also depends on proper nutrient sensing. When nutrient sensing gets dysregulated, insulin signaling becomes disrupted. Muscle cells don't respond to insulin as well as they should. They develop insulin resistance. The pancreas tries to compensate by producing more insulin. The beta cells are now overworked. They're stressed. They're exposed to high glucose levels. The high glucose causes oxidative stress and protein damage. Proteostasis fails. Beta cell senescence increases. The cells start to die off. Insulin production declines further. Diabetes develops. Obesity accelerates all of this. In obesity, there's increased adiposity. Adipose tissue is chronically inflamed. It's releasing inflammatory cytokines. This systemic inflammation damages pancreatic beta cells. It impairs nutrient sensing. It accelerates aging throughout the body. This is why obesity is associated with increased risk of diabetes, cardiovascular disease, and probably neurodegeneration. It's accelerating the aging process through chronic inflammation. Cancer is different, but also connected. Cancer results from cells that have acquired mutations that allow them to escape growth regulation and divide uncontrollably. This can happen at any age, but it becomes much more common with age because older tissues have accumulated more mutations. The mutation accumulation is partly just a numbers game. More cells dividing over more years means more opportunities for errors. But it's also because the mechanisms that prevent mutations from occurring become less effective with age. DNA repair slows down. Epigenetic silencing of oncogenes becomes disrupted. Additionally, age-related decline in immune function makes it easier for nascent tumors to escape detection and elimination. An older immune system is less able to identify and kill early-stage cancer cells. The T cells that would normally patrol tissues and find aberrant cells are depleted and dysfunctional. Senescent cells also promote cancer through their inflammatory secretions. These inflammatory factors can induce epithelial to mesenchymal transition, helping cancer cells acquire invasive properties. Chronic inflammation in older tissues creates an environment that's permissive for cancer development. Fibroblasts become activated. The extracellular matrix is remodeled. Growth factors are released. It's like preparing a garden bed for cancer seeds to grow. Metastasis, the spread of cancer, is enhanced by aging. Older tissues have compromised barrier function. Blood vessel walls become leaky. Lymphatic drainage is impaired. Older immune systems are less able to prevent cancer cells from escaping and establishing colonies elsewhere. The aging hallmarks that affect normal cells also affect cancer cells, but often in ways that make the cancer more dangerous. Cancer cells inherit mitochondrial dysfunction from their aging tissue environment, but they adapt. They become better at surviving with dysfunctional mitochondria. They shift metabolic strategies. Cancer cells acquire better metabolic flexibility. Stem cell exhaustion in older tissues means less competition for growth factors and signaling molecules. Cancer cells can thrive. This is one of the tragic aspects of cancer in aging. The same mechanisms that impair normal cell function and cause aging can actually promote cancer. It's not a simple decline into frailty. It's a complex process with perverse consequences. Many other age-related diseases follow similar patterns. Osteoporosis results from an imbalance between bone-forming cells and bone-resorbing cells. This imbalance is driven partly by inflammation, which promotes bone resorption. It's driven partly by estrogen decline in women, which impairs bone-forming cells. Parkinson's disease involves the selective loss of dopaminergic neurons in the substantia nigra. This is driven by proteostasis failure for alpha-synuclein. It's driven by mitochondrial dysfunction, particularly in cells with high metabolic demands. It's driven by oxidative stress and inflammation. Immune system aging leads to reduced ability to fight infection and also reduced ability to clear cancer. The old develop worse flu, worse pneumonia, worse sepsis. They have higher cancer rates. Slower wound healing results from stem cell exhaustion in skin and fibroblasts, impaired inflammatory response, and poor nutrient sensing in the tissues involved in healing. The list goes on. Almost every age-related disease involves some combination of the aging hallmarks. They're all manifestations of the same underlying processes. They're all results of damage accumulation, repair system failure, and network amplification. Given all of this, what can actually be done? The straightforward answer is not much, not yet. We don't have interventions that can reliably extend human lifespan in a way that's been proven in large randomized trials. We don't have a drug or procedure that can reverse aging. Aging is still, fundamentally, an unsolved problem in medicine. That said, there are some evidence-based approaches that seem promising. The most robust evidence exists for interventions that address nutrient sensing. Caloric restriction, where people eat about 20 to 30% fewer calories than normal, has been shown in animal models to extend lifespan across multiple species. Yeast, worms, flies, mice, even rats show lifespan extension with caloric restriction. It's been studied in primates with mixed but somewhat positive results. The primate studies suggest improvements in cardiovascular markers and metabolic health. In humans, we don't have lifespan data yet, because it would take decades of careful monitoring. But caloric restriction does improve various biomarkers associated with aging. It reduces fasting insulin, which is associated with reduced diabetes and metabolic disease. It improves insulin sensitivity, meaning cells respond better to insulin signaling. It lowers inflammation markers like TNF-alpha and IL-6. It improves some measures of cardiovascular function. Atherosclerosis progression slows in animals on caloric restriction. It triggers autophagy and shifts toward longevity pathways. The mechanism is through activation of nutrient sensing pathways. When the body thinks nutrients are scarce, it activates AMPK, sirtuins, and other longevity pathways. The body shifts from a growth-focused mode to a maintenance mode, to a maintenance-focused mode. The problem is that caloric restriction is difficult to sustain. Most people can't stick with it long term. It requires discipline. It requires constant attention to food intake. For many people, it's psychologically difficult. The social aspects of eating are compromised when you're always restricting. Intermittent fasting, where you don't eat for extended periods, seems to have some similar benefits. The periods of fasting trigger autophagy and activate longevity pathways. You don't have to restrict calories on average, just concentrate your eating into a smaller window. Some studies suggest intermittent fasting can improve metabolic parameters. Again, the evidence in humans is limited, but there's something interesting here. Regular exercise is another intervention with strong evidence. Exercise improves mitochondrial function by increasing mitochondrial biogenesis, the creation of new mitochondria. It triggers autophagy through AMPK activation and through the energetic demand of muscle contraction. It improves glucose metabolism. Muscles become more insulin sensitive. The ability of muscles to take up glucose without insulin improves. It reduces chronic inflammation through multiple mechanisms. Exercise reduces TNF-alpha, It shifts immune cells toward anti-inflammatory phenotypes. It reduces visceral adiposity, which is a major source of inflammatory factors. It maintains muscle mass and stem cell function in muscle tissue. Again, G athletes have better preserved muscle stem cell function than sedentary older adults. Exercise is probably one of the most beneficial interventions available, and it doesn't require drugs. It just requires consistent effort and time. As with caloric restriction, the problem is adherence. Most people don't exercise consistently. Exercise requires time. It requires effort. It requires overcoming inertia. Another avenue is targeted pharmaceutical interventions. Metformin, a diabetes medication, is being studied as a potential anti-aging drug in the TAME trial, targeting aging as the primary mechanism driving disease. It seems to activate AMPK and improve metabolic function. Metformin improves insulin sensitivity, lowers inflammation, and enhances mitochondrial function in some cell types. There's a trial underway to see if metformin can extend health span in older people who don't have diabetes but have metabolic dysfunction. Rapamycin, which inhibits mTOR, extends lifespan in mice and shows some promise in humans, though with some complications around immunosuppression, since mTOR inhibition shifts cells toward a maintenance mode, reducing protein synthesis and increasing autophagy. This makes biological sense. The problem is that chronic immunosuppression has its own costs. NAD boosters, compounds that increase levels of NAD, which is a cofactor for sirtuins, are being studied intensively. NAD levels decline with age, and NAD is essential for sirtuin function. Some small studies suggest NAD boosters might improve metabolic function and exercise capacity in older people. There's data suggesting they might improve mitochondrial function and enhance endurance in aging. Senolytics, drugs that selectively kill senescent cells, have been developed and are in early trials. The most advanced senolytics work by targeting anti-apoptotic pathways that senescent cells rely on to survive. Removing senescent cells might reduce chronic inflammation and improve tissue function. There's preliminary evidence that senolytic treatment in mice can improve physical function and reduce frailty. The evidence is still early, but intriguing. Fisetin, a natural compound found in some fruits and vegetables, has been tested as a senolytic in mice with some positive results. Others in development include ABT263 and several JAK inhibitors. Importantly, none of these interventions have been proven to extend human lifespan. Some show promise in animal models. Some show promise in terms of biomarkers in humans. But lifespan extension hasn't been demonstrated in humans yet. We're still in the early stages of aging research. The field is moving fast. New discoveries are being made. New approaches are being developed. But we haven't cracked the code yet. We haven't figured out how to reliably slow or reverse aging in humans. The future of aging research probably lies in a multi-pronged approach. One direction is senolytic drugs that can clear senescent cells. If you can remove the cells that are driving inflammation and proteostasis failure, you might be able to reset the aging clock somewhat. There's evidence in mice that removing senescent cells can improve physical function and extend health span. The challenge is doing this in humans without causing harm. Another direction is restoration of proteostasis. If you could enhance proteasome function or enhance autophagy, you might be able to clear the protein aggregates that accumulate in aging. This could help prevent neurodegenerative disease. There's research on drugs that enhance autophagy. There's research on drugs that enhance proteasome function. This is more difficult than it sounds because these systems are finely balanced. Increase one too much, and you can cause problems. But the potential is enormous. Mitochondrial restoration is another avenue. If you could improve mitochondrial function in aging cells, reduce free radical production, enhance energy production, you might be able to address multiple aging hallmarks at once. This might involve enhancing mitochondrial biogenesis, the process by which cells make new mitochondria. It might involve targeting free radical scavenging. It might involve restoring the function of damaged mitochondrial proteins. Telomerase activation is another potential approach. Telomeres shorten with each cell division. When they get too short, cells enter senescence. If you could reactivate telomerase, you might be able to extend cellular lifespan. The risk is that this could promote cancer, because cancer cells often use telomerase to achieve unlimited replication. But there might be ways to do it safely. Systemic rejuvenation through parabiosis, where blood from young animals is transfused into old animals, has shown some promise in animal studies. There are circulating factors in young blood that seem to promote regeneration in old tissues. Identifying these factors and replicating them pharmacologically could potentially provide systemic benefits. Finally, there's the possibility of more fundamental approaches. If aging is fundamentally a problem of accumulated damage, maybe you could use gene therapy to enhance DNA repair machinery. Maybe you could use epigenetic reprogramming to reset epigenetic patterns to younger states. These are more speculative. They're further from clinical reality, but they're being researched. The field of aging research is in a state of rapid development. What seemed impossible 10 years ago is being investigated today. What seems speculative today might be clinical reality in 10 years. We're at the beginning of understanding aging at a molecular level. We're at the beginning of being able to intervene. In the end, aging is still a fundamental biological process that we're only beginning to understand. The mechanisms are complex. They're deeply interconnected. They involve systems that evolved for reproductive success in ancestral environments where scarcity was real. They involve the accumulation of damage over time through oxidative stress, radiation, and errors in molecular machinery. They involve the gradual failure of repair systems and the exhaustion of cellular maintenance capacity. They involve the amplification of problems through feedback loops, where damage causes more damage. We've identified some of the hallmarks of aging. We're beginning to understand how they interact. We're developing tools to target some of them. But we don't have a cure yet. We don't have a pharmaceutical silver bullet that can stop aging or reverse it in humans. What we have are hints. We have evidence that some interventions help a little. We have understanding of mechanisms that might be targeted. We have hope that further research will lead to better approaches. And in the meantime, the best evidence we have suggests that the most reliable ways to improve health span are still the boring ones. Move your body regularly. Don't eat constantly. Sleep well. Reduce stress. Maintain social connections. These are old advice, but they're still true. They work because they address multiple aging mechanisms simultaneously. They reduce chronic inflammation. They improve mitochondrial function. They activate longevity pathways. They enhance autophagy. They maintain stem cell function. They're not glamorous. They won't make headlines. But they work. And while we wait for the breakthroughs in senolytics and mitochondrial restoration and epigenetic reprogramming, the most powerful tool you have against aging is probably still just the life you lead, the choices you make, the movement you do, the food you eat, the sleep you get, the people you connect with. These things matter. They slow aging. They improve health span. They buy you time. And perhaps, if the aging research continues to progress as it has, that time you buy through living well might eventually overlap with the time that new interventions become available. You might slow aging through your lifestyle long enough to benefit from drugs that slow it even further. You might extend health span through exercise long enough to see senolytics become available. The future of aging is still being written. And while we can't stop it yet, we're getting closer. We're learning. We're trying.